Transcriptomics is defined as the study of the transcriptome, which is a complete set of RNA transcripts that are present in a specific tissue or a culture, cell, a specific point in time, and under a specific set of circumstances. Typically, this includes both the qualitative and quantitative information, which may be a transcript structure as well as the abundance of it. There are several different kinds of RNA species, which may be rRNA, tRNA, and mRNA, which are the best characterized. There are also several other types of RNA species with varying degrees of specificity. For example, miRNA, the long non-coding RNA, the RNAseq, and etc. There are several reasons why transcriptomics is a powerful tool in biology. First of all, RNA is a chemically homogeneous compound, a chemical compound, because proteins contain, for example, 20 amino acids, like combinations of these 20 amino acids, while RNA has 4 nucleotides. It is therefore technically much easier to analyze many different molecules of RNA. A crucial technical advantage of working with RNA or even DNA are its hybridization properties, which can be exploited in sequencing reactions. In contrast, proteomics is new, but it is very difficult to apply to complex samples with many proteins. It is also more expensive and less sensitive and less quantitative as compared to RNA. Gene expression has also provided numerous insights into biological processes in the past. So transcriptomics is a very useful way to study the internal functioning of a cell, tissue, or culture. After we isolate RNA, there is usually an additional selection step carried out to enrich the sample for the particular type of RNA that we are interested to study. For example, we focus on mRNA, and that involves the removal of rRNA and tRNA. So without selection, more than 90% of the sequencing data would be irrelevant for us. We perform selection procedures like poly-A selection, which is a long chain of A. This is a very effective way of removing anything but the mRNA and some minor RNA species with a poly-A tail. For example, the long non-coding RNAs. However, if we are specifically interested in the long non-coding RNAs, not all of them will be captured this way, as not all of them have a poly-A tail. Another thing to remember is that bacterial mRNA do not have a poly-A tail. So here the rRNA depletion becomes the only option. The rRNA depletion is the second technique to isolate the RNA. Here we have a more indirect way of increasing the abundance of mRNA in the sample by taking out the rRNA in using some probes that are covered in a bead. We can also have a size selection. By selecting small RNAs, the sample can be enriched for the smaller length of RNAs, for example, the miRNAs. Once we do this, we will be able to construct a library out of the RNA. So the steps that are taken to do that are, first of all, we fragment the RNA. Then we synthesize the cDNA out of it by using a pool of primers with 6 random nucleotides that are the random hexamer priming. So each sequence can be reverse transcribed. Third, we can add the sequence adapter by using the primer which overhangs or by ligation. And finally, we can amplify using PCR. This appears similar to genome sequencing, but with RNA, we often try to remember the strand specificity, which means which particular gene is attributed to what strand of the RNA. This is important, especially in cases where genes overlap, but are transcribed from the opposite direction. This allows us to determine which reads have attributed to what gene. When we do not do this, we lose a layer of information. So this is accomplished by using certain tagging sequences during the time of annealing with the synthesis of the DNA for the particular RNA template. Now we can cover the topic of sequencing, which means that now with the DNA sequences that we have and not the RNAs, because we have to keep in mind that we already converted it into a cDNA. The sequencing is done using similar technologies as is done with the next generation genome sequencing. So we can create a bioinformatic workflow where we begin with the raw reads data, which may be in a .fastq file, and then we can expose it to quality control and some pre-processing, which can give us the pre-processed reads, and then we apply a mapping strategy that is provided by Bowtie, Top Hat, or some BWA software tool that helps to align the reads to the genome or transcriptome if a sequence genome is unavailable. So this is called the mapping strategy. If we do not have a sequenced genome, then we will have to assemble the entire DNA sequence using a de novo transcriptome assembly, where the transcripts are assembled without any prior knowledge by comparing the sequences of the reads and then making contigs. Basically, the overlapping reads are joined together to construct transcripts and generate a representation of the transcriptome. Once we have mapped the reads, we generate a file which may be in the format of a .bam file or a .sam file. This is exposed to the stage of transcript discovery. So a software like Cufflinks will have features that assembles the transcriptome and then compares it to the reference annotation. The differences between these representations indicate regions containing new transcripts, possibly. It is recommended to always manually check the new assembly before using it in the next step where we count the reads. For example, we can look in IGV if the newly annotated transcript looks at all like the well annotated ones before we add it to the reference annotation. In short, basically how we are defining the transcripts from the mapped short reads is that we generate a coverage contigs that are built from positions where the short reads are mapped above a given threshold. Then a list of candidate exons is defined for each coverage contig by searching for splice sites that may be 100 nucleotides around the boundary, and then we can use the unmapped reads to select the correct junctions, and then the exons are linked to construct the whole transcript together. This last step starts from differently linked exons and uses the detection of coding sequences to distinguish the real transcripts. The output of this step gives us a .gtf or a .gff file, which we can send to the next stage, which is the final step of counting and statistically testing the data that we have. It may involve software such as CuffDiff, HTSeq count, Lemma, DSEC2. This stage is begun by counting estimating the expression level of the gene. So this expression estimate can be compared for different conditions. Or simply put, there are two steps. First, we count the number of reads that fall in a certain interval, that is a gene or an exon, and then we test if there is a significant difference between these numbers for different conditions. So a software like HTSeq, which counts the reads, employs a simple principle of counting how many aligned reads map to each predefined feature or an interval, which we said before might be a gene or an exon. There are different ways of dealing with reads that do not completely fall within one feature. For example, there may be one read which can be completely composed within a gene A, or it may be only partially composing a gene A and partially sharing another gene region. So we try to create an HTSeq table where we have a probability template for union, intersection, or a non-intersection. So we don't need to get into the detail of that, but they have a specific decision scheme to determine how the reads might be annotating a particular interval. Quite often, we will be interested to study and compare the gene expression between two different conditions. So using the raw count data as an input for these tests is not a good idea because they might have a certain number of biases. That means that the different samples may not be able to may be unable to compare just with the raw counts. So we can try to normalize our data. We can find the most common sources of bias in three particular places. For example, the sequencing depth. The total number of reads will always differ between two samples. This is due to the quality of the RNA or the cDNA, or even the imperfections during the library constructions. So, if a library has a different size, the gene counts cannot be compared directly. For example, let's say that gene X has 20 reads and in sample A and 10 in sample B. So, if these numbers are compared without further context, the conclusion would be that gene X has a twofold higher expression in sample A. However, if sample A would have a total library size of 200 reads and sample B of 100 reads, then it is easy to see that the expression of gene X is actually the same in both the samples. Another source of bias is the gene length. When comparing the gene expression within one sample, the gene length is something that can vary. For example, gene X has 20 read counts and gene Y has 10. Then a naive assumption would lead to us saying that gene X is twofold higher expression than gene Y. But suppose that gene X has a length of 2 kb and gene Y has 1 kb. Then for gene X, 20 reads will be distributed over 2 kb, which equals the distribution of reads for gene Y. That means that both the genes have similar expression. So in general, longer transcripts will give rise to more reads than shorter transcripts with a similar expression. The third situation of bias may come from the differences in count distribution. Sometimes a small number of highly expressed genes can take up a large portion of the total amount of reads, thus reducing the confidence with which the decisions can be made for the remaining number of genes. A change from 1000 to 2000 reads is more statistically significant than a change from 1 to 2 reads. So the total amount of reads is limited, and if some genes are highly expressed, they will increase the uncertainty for the remaining ones. So we always try to normalize our count data using the information of the total count and the particular gene. Now that we have our data that is normalized, we can try to compare the two conditions that we have. For example, using an MA plot, we can observe the tendency of particular genes that are expressed differently in different conditions. If a gene in condition A has a normalized count of 100 and in condition B it has 200, then we can say that the gene has a fold change of 2 or a log 2 fold change of 1. For observing this, we can make a plot as you can see in the screen right now. In the MA plot, the M values are the log of the gene count ratio between the samples, and they are plotted against the abundance of the normalized counts. The MA plot can highlight the outliers and also show the deviation between the gene counts. So, both in the sense of individual genes or the whole samples. The plot typically has a large spread at the lower level of abundance due to the Gaussian sampling error that is dominant in this range. For example, it is possible that a gene only has 1 count in condition 1 and 8 counts in condition 2. The apparent log fold change would be 3 because log of 8 to the base of 2 is 3. But this is clearly due to the random effect at this low level of count. So for low expression values, there will be many genes showing this pattern. In order to see the differences between two particular conditions, the genes that pass the significance threshold and are considered differentially expressed are colored red in this particular graph. The black line highlights the overall mean, which shows no fold change in that case. And the values are symmetrically distributed across the mean, across the abundance range, with no strong biases. The higher variability at the low ranges highlight the Gaussian sampling noise, which is dominant there, as we said before. Finally, we can cover the sources of error for an RNA-seq data. So initially, there might be some variability between the transcripts. This is a stochastic effect that occurs purely due to chance, which will cause variability. Just like when we count the heads or tails after tossing a coin, the sum will vary even with a perfect coin. So, this type of variability can be sizable and even dominant for low abundance transcript, and that is why it is called short noise or Gaussian noise. A way to deal with this noise is to increase the size of your library and use more material to generate them, that is, more reads for the deeper sequencing. The second source of variability may be technical, that is, the variability caused by the sample preparation and the sequencing. This is the kind of error that is controllable and can be reduced by focusing on better laboratory practices like a tighter control on the process, higher quality chemicals, the choice of sequencing technology, etc. Third source of variability has its origin in the biological samples. So the biological variability or some biological noise is dominant for higher expressed genes. The variability between cells and samples is an inherent feature of biology, which is unavoidable at some point. So it is crucial to think very carefully about the experimental design to limit this variability. So, for numerous studies, we can limit this effect by using some exponential growth of some bacteria or the stationary phase or choosing the point of data collection carefully.